In this lesson, we are looking at variation through sexual reproduction. So we're still talking about meiosis, we're just looking at a more specific part of it. One of the benefits of sexual reproduction is that the organism inherits a new, unique combination of genetic information, and this increases the diversity within a species, which we know is beneficial for the survival of a population. Now, sexual reproduction and in turn meiosis increases the possibility of variation within the new generation in three main ways. There's recombination and crossing over of chromosomes in prophase 1. There's independent assortment of paternal and maternal chromosomes in metaphase 1, and there's random fertilization of gametes. And we're going to go through each of these separately. So right at the beginning of meiosis in prophase 1, there are homologous chromosomes coiling up, getting all intertwined, and they form bivalents. This is a bivalent here, right? The reason these bivalents form is to increase the possibility of gene sharing. When they are hugging really tightly, the arms of the chromatids are able to cross over. And because homologous chromosomes have the same genes, just different varieties of them, they're in the same sequence, they can scramble the paternal and maternal genes and rearrange the combinations of gene variants on each of those chromosomes. This is why it's called recombination or crossing over. Now, the specific region where the sister chromatids from different chromosomes interact or cross over is known as a chiasma, right? So it's this crossing over point there. You can see that in this situation that the type of gene hasn't changed, right? We've still got the D gene, it's still in the same place. It's just the variant of the gene has changed from one chromosome to the other, right? So from the capital D to the small d. And it's only actually happening on one chromatid. So when these chromosomes undergo meiosis and split into different gamete cells, you can see that it's going to increase the variation amongst the gametes. They're no longer just containing only maternal or only paternal information. There's a combination ready for inheriting. The second way that variation is introduced in meiosis is through metaphase 1 via independent assortment. So again, this is really early on. In fact, this is directly after recombination has occurred in prophase 1. Independent assortment is going to happen. And in metaphase 1, the chromosomes line up with their homologous partner at the equator of the cell. And we know that one pair will head in one direction and one will go in the opposite direction. But it's because all the pairs line up at random that it's very little chance that all of the maternals are going to go one way and all of the paternals are going to go in the other direction. So if you look at this example, it's only got two types of chromosomes or two pairs of homologous chromosomes, the big ones and the small ones. They've tried to use the color coding, so pink is maternally inherited, blue is paternally inherited, gender roles and colors. So we can see they're going to line up, but we've also got another option. If they line up in this fashion, these are the kind of gametes we'll end up with. But if they line up like this, then we're going to end up in this situation with all paternal and all maternal on different sides, and then the gametes are going to look different. So consider this with your 23 pairs of chromosomes instead of just two, as well as the ability to do the recombination beforehand, and the combinations are immense. And this is why it's called assortment. Each gamete will receive a completely random mix of maternally and paternally inherited chromosomes. So we're still only going to get one of the homologous pairs because we know we're only trying to make a haploid cell. But this is increasing the possible combinations of genetic material available in the gametes. This is on top of the variation increased already at the recombination stage. Now, the final way that variation is ensured in sexual reproduction is through the actual gamete cells during fertilization. Now, in essence, it ensures that any gamete with any random combination of chromosomes can participate in fertilization, and therefore the number of possible combinations of genetic traits in the offspring is going to be extremely large. If one single cell can have one of 8 million possible combinations, and it's fertilized by a sperm with 1 in 8 million possible combinations, it provides even more possibilities of combinations of genes in the new offspring. So while you and your siblings may inherit their genetic information from the same people, you know that the immense amount of possible combinations of genes inherited by you will never be the same as another, right? So you will have the same resemblances maybe, but you can't produce the exact same unique combination unless you're an identical twin, which is a story for another day. Okay, so again, we're looking at meiosis still, but now we're looking specifically at the variation that meiosis can create.